again. So we have Jessica's talk on YouTube.
Yes, sorry. Okay.
All right.
We're good.
Yeah, we're good. So let me restart introducing Dr. Jessica Stark. Jessica did her PhD at the Northwestern University, and she is currently a postdoctoral fellow in Dr. Caroline Bertozzi's lab at Stanford University, funded by the American Cancer Society. She is going to tell us about some of the really cool state-of-the-art technology development she completed targeting this really complex but extremely important glycobiology side of our immune system. So please take over, Jessica.
Thank you so much for that introduction, and thanks so much to Leading Edge for this opportunity to share my work. Engineering the immune system through the use of vaccines and immunotherapies has the potential to provide cures for human disease as well as completely eradicate certain pathogens. However, in light of emerging pathogens with pandemic potential, as well as limited patient responses to existing immunotherapies, I think this highlights the need to develop additional interventions to address unmet medical needs in autoimmunity, infectious disease, and cancer. And one understudied and under-targeted area of immune regulation has to do with sugars. Sugars, or glycans, make up these glycoconjugate molecules that coat the surface of every cell and extend far into the extracellular space, providing one of the first points of contact with surveying immune cells. And even more interestingly, the chemical structures of these glycoconjugates are distinct between different organisms, as well as can be altered in the context of disease, representing both organism and disease-specific antigens. And perhaps because of this, glycans are further involved in non-self discrimination, acting as ligands for immune checkpoints that can either activate or dampen immune responses. And these can further be altered in the context of disease. So in light of these profound roles that glycans play in terms of modulating immune responses, my work has focused on better understanding how glycans modulate the immune system and then targeting these molecules for vaccines and immunotherapies. And so today I'm going to tell you two stories about how we've used or targeted glycans to create vaccines against bacterial pathogens, as well as new cancer immunotherapies. So I'll begin with vaccines. I think the SARS-CoV-2 pandemic, as well as some of the talks that we'll hear today, really highlight that infectious disease is far from a solved problem in our modern times. And in particular, I'm here to tell you that drug-resistant bacteria represent a growing global concern. These drug-resistant bacterial infections are predicted to threaten 10 million lives per year by 2050. And at this point, drug-resistant bacteria will outpace cancer as a leading cause of death worldwide. And importantly, the burden of these drug-resistant bacterial infections will be predominantly felt in lower middle income countries. So not only do we need new interventions for bacterial infections, but we also need to be able to distribute these worldwide. Conjugate vaccines are a class of FDA-approved molecules that represent one of the safest and most effective ways that we have to combat drug-resistant bacteria. These molecules are actually glycoproteins. They use bacterial cell surface glycans as antigens to elicit cellular and humoral immunity to bacterial pathogens. And to do this effectively, we conjugate these glycans to an immunostimulatory carrier protein. And just to illustrate the efficacy of these molecules, I'm showing you data for Haemophilus influenza infections, both before and after introduction of a conjugate vaccine that targets this pathogen. And you can see that the vaccine has dramatically reduced the rates of infection. And because of this, it's also reduced reliance on antibiotics to treat infections, which can slow the rate of emergence of drug-resistant forms of the bacteria. So despite the efficacy and safety of these molecules, we only have a few that are approved by the FDA and part of standard immunization regimens worldwide. And I think there are two key reasons for this. The first being that these molecules are really difficult to both develop and produce from a technical perspective. And further, they're difficult to distribute, requiring cold chain for distribution. And as a result, some of these highly effective vaccines, their vaccination rates are quite low, on the order of 30% worldwide. So we thought perhaps if we could develop new ways to both develop new vaccines and distribute them, we could enhance our ability to use conjugate vaccines to prevent drug-resistant bacterial infections. And so during my PhD, I developed a cell-free system that allows us to rapidly produce and distribute conjugate vaccines. Our cell-free system uses cell lysates rather than living cells to produce these vaccines. And we showed that in the context of this cell-free platform, we were able to rapidly produce a variety of different conjugate vaccines targeting different pathogenic bacteria of interest, including a couple of strains of pathogenic E. coli, as well as Francisella tularensis, which I'll talk more about in a moment. We further showed that the cell-free lysates could be freeze-dried for distribution without the need for cold chain. And then once they arrive at the point of care, they can be reactivated to rapidly synthesize vaccines by simply adding water to rehydrate the reaction. And in the context of this portable vaccine production and distribution platform, we showed that vaccines against the highly virulent bacterial pathogen Francisella tularensis could completely protect mice from lethal challenge with this bacteria. And in fact, were just as effective as vaccines produced using an existing technology. So overall, we think this will really allow us to rethink how we both develop and distribute conjugate vaccines against emerging pathogenic bacteria. So I'll switch gears now to talk about how we can target glycans for cancer immunotherapy. It turns out that cancer cells exhibit strikingly different cell surface glycosylation compared to healthy cells. And one example of this is that tumor cells upregulate a particular glycan on their cell surfaces called sialic acid. It's actually been long understood that tumors do this. This is a phenomenon that's been observed since the 50s and 60s and in both solid and liquid tumors, but it was a real mystery why tumor cells would upregulate sialic acid in this way. Until about a decade ago, through genome sequencing efforts, it was discovered that there's this class of sialic acid binding receptors called Siglecs that are expressed broadly across different cell types in the human immune system. There are 14 of these Siglec receptors in humans, and interestingly, 8 of them bear striking homology to the established immune checkpoint PD-1. So just like when PD-1 binds to its ligand PD-L1, when Siglecs bind to these sialylated glycans on tumor cells, they recruit phosphatases to the immune synapse through ITIM and ITSM domains, which dampens immune activation and is thought to allow tumors to evade detection by the immune system and resist therapy. And there's now a growing body of literature that suggests that these Siglec sialic acid axes play key roles in tumor progression and represent attractive targets for immunotherapy. So as you might imagine, there's great interest in creating new immunotherapies to target this axis. And in particular, the ligand side of this interaction, so the sialylated molecules on tumor cells, have been particularly difficult to target in a very selective way, representing an open sort of area for drug development. And perhaps the reason why not much progress has been made in this area is because existing therapies sort of fall short when it comes to targeting these glycans. Monoclonal antibodies are sort of a standard in the field for targeting the ligand side of these immune checkpoints because they can have high affinity for tumor-associated antigens. However, it's been a long-standing technical challenge to develop antibodies that can bind to sugars or glycosylated molecules with high selectivities and affinities. Another approach that people sometimes use is to develop these decoy receptor type molecules, where they take the soluble ectodomain of the checkpoint receptor of interest and fuse it to an antibody FC. And this approach has the advantage of retaining all the specificity and selectivity for the ligands of interest, and the same selectivity and specificity that the endogenous receptor would have. However, these molecules, at least for glycan binding receptors like Siglex, their affinities are just too low to be therapeutically relevant, on the order of micromolar or low millimolar affinities for their ligands. But we thought perhaps we could combine the beneficial properties of both of these molecules into these antibody lectin bispecifics that we're calling ABLEX. The antibody side of our molecule would recruit this protein to the tumor cell surface with high affinity and selectivity by targeting a tumor-associated antigen. And then at that point, the molecule would accumulate at high enough local concentration to permit binding of this otherwise low affinity glycan binding domain from the targeted receptor, allowing us to rescue this sort of decoy receptor approach for checkpoint blockade. So that was our hypothesis. We made these molecules and tested their ability to bind to cells. And I think this really illustrates the advantage of our bispecific platform. So compared to the decoy receptor-like molecules, which we made from 2 inhibitory Siglex that have been shown to play roles in tumor progression, Siglex 7 and 9, these decoy receptor molecules just don't bind to cells at therapeutically relevant concentration. But if we take those same decoy receptor arms and combine them with the high affinity tumor targeting antibody arm from trastuzumab, which targets the tumor-associated antigen HER2, these bispecifics bind to cells and exhibit low nanomolar dissociation constants, very much on par with the parent monoclonal antibody. We next tested whether these molecules could enhance anti-tumor immune responses, the thought being that in the context of monoclonal antibody therapy, if the Siglex-sialic acid axis is engaged, this would dampen immune responses to our tumor and allow tumors to resist treatment. But if we could create the same tumor targeting effect with our bispecific while simultaneously blocking the Siglex axis, perhaps we could potentiate an enhanced anti-tumor response. And we tested this in the context of antibody-dependent phagocytosis and cytotoxicity assays in vitro, where we use human immune cells expressing Siglex 7 and or 9, and feed to them tumor cells that express ligands for those receptors, as well as the targeted tumor-associated antigen HER2. And what we think is happening is that indeed this Siglex axis is suppressing antibody effector functions, because when we treat with our bispecifics, we're able to further enhance tumor cell killing by phagocytosis or cytotoxicity compared to controls, including the monoclonal antibody in purple. We think this platform has the potential to be really modular for targeting multiple glycan-based checkpoints as well as tumor-associated antigens. We made different bispecifics that combine the Siglex 7 decoy receptor as well as antibodies targeting alternative tumor-associated antigens. The rituximab antibody targeting CD20 antigen, and the cetuximab are antibodies targeting EGFR. And we showed that these molecules, just like in the context of our trastuzumab hybrids, did enhance phagocytosis and killing of tumor cells compared to the parent antibody. We're looking now to prove this concept in vivo, as well as expand to target additional glycan-based immune checkpoints and tumor types. So to summarize, I hope I've convinced you that glycans play central but underappreciated and understudied roles in immunology. And through the development of new biotechnologies, I've been able to chip away at some of these fundamental questions about how glycans regulate the immune system and translate that knowledge into the development of new therapies. My future research group will continue to innovate in these 3 synergistic areas. And one kind of overarching question, some of the overarching questions that we'll ask is how glycans influence different immune phenotypes that we observe, whether those be pro or anti-inflammatory, and how we can engineer those phenotypes in the context of diseases like autoimmunity and cancer. So with that, I'll just thank my postdoctoral and PhD advisors, my collaborators, my funding sources, and thank you all for your attention. I'm happy to take questions.
Thank you, Jessica. That's quite amazing. The translational power is like limitless. So we can wait for people to type their questions if they have any. I have many questions. If it's allowed, I was wondering, in the context of host immune interaction, no, host pathogen interaction, how adaptive are the pathogens in reshaping these glycans? Because usually their cell wall sugar declaration can be modulated through both mutations and also gene expression regulation, right? Like, what's the how big is the danger of immunization, and how I know that your system is pretty modular and can be easily adapted to different iterations, but how well do the bacteria do in this?
I see. Well, let me answer one question first. I think one of your questions was how does infection kind of change glycosylation patterns? And I think kind of the most well-studied area is in the context of viral infections. Viral infection is known to kind of change glycosylation patterns. And in particular, I think the best studied cases are in the context of HIV infection, where infection is kind of correlated with these highly monosylated glycan structures. And in fact, I think a lot of work has been done showing that these shield viral epitopes from the immune system, but this could also be involved in immune modulation. And some of these monosylated structures have also been shown to facilitate uptake by APCs through DC sign or other mannose binding receptors on those immune populations. So I think this is a really interesting area. And there's still, while we know some things about how viruses modulate glycosylation, I think there's still a lot to learn, and potentially we could pull out some interesting viral associated antigens that have to do with glycans. Again, in the context of HIV, it's been shown that some of the broadly neutralizing antibodies are actually glycan reactive. And then your other question was about kind of how does glycosylation change in the context of bacterial infection, or what's the potential for immune escape by bacteria? So I think the best understood examples of that are in the context of existing conjugate vaccines. We know that in fact, even different strains of the same bacteria, as well as from different kinds, across different kinds of bacteria, you see different cell surface glycans being produced. In existing vaccine formulations, you have multiple glycoproteins, multiple conjugate vaccines that are decorated with those different sugars to allow broad protection against all of those strains or all of those pathogens. But we haven't seen a lot of immune escape. These conjugate vaccines remain very effective despite being in use for several decades. So I think, just like viral vaccination, sometimes you have to capture many different strains in the initial formulation, but escape seems to be not an incredible worry. And yeah, I think that's an advantage of our platform, actually, that we're able to really rapidly develop these different proteins that are modified with different sugars so that we could co-formulate.
Yeah, that's really good to hear. And Jeanette asked, for the cancer targeting bispecific antibodies, have you thought about changing the isotope to change the valency?
Yes, that's a great question. We have thought about that in the context of perhaps we could increase the valency of our glycan binding arm to make it higher affinity. And the reason why we're interested in that is it would be really interesting to make sort of an AND gated or logic gated bispecific that requires both the tumor associated antigen that's targeted by the antibody like HER2, CD20, EGFR, as well as expression of these ligands for Siglex for binding, because that could give us additional selectivity for tumors and kind of minimize these on target off tumor effects that people sometimes see with immune therapies. So we're not necessarily thinking about changing the isotype to something like IgM, but what we have been thinking about is perhaps engineering additional glycan binding arms through C-terminal fusions or of the heavy chain or light chain to kind of increase the valency. We've also been thinking about affinity maturing the glycan binding domain to see if we could make it higher affinity.
All right, thank you so much, Jessica. Let's move on to our next speaker. Our next speaker is Dr. Chinyere Iweka. Chinyere did her PhD at Georgetown University, and she is currently a postdoctoral fellow in Dr. Katrin Anderson's lab at Stanford University. And congratulations to Chinyere too, who recently received a Burroughs Wellcome postdoctoral fellowship. Yay. So Chinyere is gonna tell us about some really interesting discovery she made on the circadian rhythm regulation and aging on stroke severity. Thank you, Chinyere.
Thank you so much for the kind introduction, and thank you to Kara and the Leading Edge team for giving me the opportunity to share my research with you today. So I'm going to take you a little bit away from infectious diseases and ask you this very important question. Did you know that so I'll begin with a question. Did you know that a stroke occurs every 40 seconds and a resulting death every 3.5 minutes in the United States alone? With aging population increasing, stroke incidence is expected to dramatically increase. The estimated cost for stroke between 2017 and 2018 was $53 billion and is projected to be $1.5 trillion by 2050. Thrombolytic therapy and embolectomy are the only current interventions for stroke patients, with a very narrow intervention window of 4.5 to 16 hours, benefiting less than 10% of stroke patients. Therefore, there's a critical need to understand the stroke process and identify effective therapies that reduce stroke severity and improve outcome. So I'll begin by telling you about the stroke process. First, a stroke occurs due to a blockade of cerebral arteries, preventing oxygen supply to neurons, and that the dead and dying neurons will release these danger-associated molecular patterns that will leak into the peripheral circulation, and this activates the innate immune system. And then these cells will transverse into the brain parenchyma. But instead of a resolution, these cells initially release a cytokine storm, an amplified inflammatory response that further exacerbates the stroke injury. So my question is, why do immune cells initially contribute to the stroke injury? Well, first, in order to mount a healthy immune response, a homeostatic metabolism is required. Several studies have established that immune cell function is dependent on its metabolism, and other studies have established that metabolism is impaired with aging, a non-modifiable risk factor for stroke. Well, how is metabolism regulated? It's tightly regulated by the circadian clock. Shown here in this illustration are core clock proteins, BML and clock, as well as PERT and CRY, regulating levels of NAD, a critical energy cofactor essential for metabolism. Indeed, NAD has been shown to exhibit rhythmicity in its levels, and deletion of core clock protein BML has been shown to significantly reduce NAD levels in the whole cell and in the mitochondria. Immune cells also exhibit rhythmicity. In this study by Nguyen in 2013, they showed that blood monocytes exhibit rhythmicity in their expression of core clock protein BML. Lysix high monocytes, which are pro-inflammatory monocytes, exhibit rhythmicity in their numbers in the blood and in the spleen, as well as in their expression of pro-inflammatory molecules. This is in anticipation of immune challenges throughout a 24-hour period. Another study by Laura Funkin in 2015 showed that microglia, the immune cells of the brain, exhibit rhythmicity in their expression of core clock proteins, as well as in their expression of pro-inflammatory molecules. So we have well-established studies that have demonstrated diurnal variation in immune cell function and in metabolism. So the question now is, what is the circadian clock? Well, I'll tell you a little bit about it. It is a clock function that functions to allow temporal coordination of all the physiological processes within an organism and its external world. Here is the suprachiasmatic nucleus. It's a pair of tiny nuclei that is the master oscillator and is located in the hypothalamus. And it is entrained by feeding schedules, light dark cycles, and activity. And it synchronizes all of this with the clocks in the different brain regions as well as in the different organs. This is to maintain homeostatic processes. This way, all of your organs are working in concert to anticipate changes in the environment. While there's organ level synchronicity, there's also cell autonomous regulation, where 15% of all genes are rhythmically regulated by the core clock protein shown here. And I told you briefly about BMO in clock, purr and cry, roar and rev ebb. Disruption of the circadian clock has been linked to several diseases. You can disrupt the circadian clock by social jet lag or by shift work, where the times you're working days and nights and eating are constantly changing. And this has been linked to chronic health conditions such as cancer, cardiovascular disease, and metabolic disorder, as well as shown in this meta-analysis in 2020. Circadian disruption has also been linked to increased stroke incidence, as shown in this study in 2009 of nurses that do shift work. And circadian disruption has been well established to have immunological effects. Additionally, with aging, circadian rhythmicity is disrupted by either being completely lost, dampened, or shifted, or genes that were formerly rhythmic, not rhythmic, begin to exhibit rhythmicity. Shown here in this illustration, when you compare young adults in blue and older adults in red, are some of the activities that you can see are shifted with aging, including waking activity, SCN firing, temperature levels, cortisol levels, melatonin, and plasma glucose levels. So my hypothesis is that the disrupted circadian clock in aged individuals results in impaired metabolism that causes a maladaptive immune response to all kinds of diseases, but specifically in my research, to stroke. So to begin to address why immune cells have a maladaptive immune response to stroke, first I wanted to assess the metabolic status of these cells before they even get into the brain, since their function is dependent on their metabolism. To do this, I used young and aged mice because I wanted to understand the differences in stroke in response to age as a factor. I subjected the mice to middle distal cerebral arterial occlusion. I collected the blood monocytes and then measured oxygen consumption rates. I chose 3 different time points to identify metabolic changes that are constantly changed over time and might provide insights into the metabolic stroke response. Through this method, you can determine basal respiration, ATP production, maximal respiration, as well as other parameters. So interestingly, at 4.5 hours after stroke, the blood monocytes from young sham and young strokes show no differences in their oxygen consumption rates. But when you look at the aged stroked mice, you can see their blood monocytes have increased oxygen consumption rates. When we look at basal respiration, maximal respiration, and ATP production, in the young, they show no differences. But you can see a significant increase in these parameters in the aged mice, highlighting age as a factor in the stroke response. At 24 hours after stroke, oxygen consumption rates are now significantly decreased in the young strokes, as well as in the age. In the age, initially they were high, significantly higher, but now they have significantly decreased. And when we look at the previous parameters, basal respiration, maximal respiration, and ATP production, you can see that all of these parameters are significantly decreased both in the young and in the aged mice. This suggests metabolic exhaustion at this time point. By 72 hours after stroke, the oxygen consumption rates of these blood monocytes from the young have now rebounded to sham levels, whereas in the aged, they remain metabolically depleted. And when we look at the same parameters, you can see no changes in the young, but in the age, they're significantly decreased. So altogether, these results suggest that age is a factor in a metabolic response to stroke, as you can see here in these different traces in the young and in the age, and therefore could be targeted to improve stroke outcome. So my next question was, what specific metabolic pathways change in response to stroke before these cells enter the brain? I subjected the mice again to middle distal cerebral artery occlusion, collected the blood monocytes, and then performed on-targeted metabolomics using liquid chromatography, mass spectrometry. Many metabolites of different pathways were detected, but for this talk, I will focus on NAD, which is the critical energy cofactor, and quinonic acid, which is a metabolite that is converted to NAD, but is also a neurotoxin, and it causes excitotoxicity when it accumulates in neurons. So at 4.5 hours after stroke, when you compare the young sham to young stroke, you can see no differences in NAD levels at this time point, but you can see a significant increase in quinonic acid. In the age sham, they have increased NAD levels, and quinonic acid is also elevated but did not reach significance at this time point. By 24 hours after stroke, you can now see that there's increased NAD levels in these blood monocytes from the young stroke. Quinonic acid remains elevated, and in the age, you can see now a significant drop in NAD levels, while quinonic acid remains elevated. By 72 hours after stroke, NAD levels have rebounded to sham levels, while quinonic acid is also rebounded. But in the age, while NAD levels are coming back up to sham levels, quinonic acid remains elevated. So the results so far show that blood monocytes in aged and young mice have very different energy patterns. When you look at the NAD traces, the green traces here in the young and in the aged, in response to stroke, and these cells are already producing a neurotoxic molecule before they even enter into the brain. The next question I asked was, how does disrupting the circadian clock impact metabolism of blood monocytes before they enter the brain after stroke? Again, I subjected the mice to chronic jet lag first by shifting the light-dark cycles every 3 days by 6 hours for 4 weeks. And then I stroked the mice and measured oxygen consumption rates after 24 hours. Comparing young control stroke to jet lag stroke, shown here in red and in purple. And this cartoon here is just an illustration of the results here to make for better clarity. You can see a significant drop in oxygen consumption rates in the jet lag stroke mice. However, when you compare the control sham to the jet lag sham in green, you can see an increase, but this did not reach significance. So these results indicate that circadian disruption has an effect on the immune cell metabolism and the response to stroke. So altogether, my results indicate that age is a factor in the immune response to stroke injury. Comparing young and aged mice, you can see very different metabolic responses, showing very different energy patterns. The patterns of NAD levels in the blood monocytes from the young and aged mice are also very different and is consistent with the energy demands at the time points investigated. And they both have quinonic acid, very high quinonic acid levels, a neurotoxin before they even enter the brain. And finally, disruption of the circadian clock has a significant effect on the metabolism of blood monocytes, impacting the immune response to stroke. Given that circadian rhythmicity is disrupted with aging, I'll be repeating the jet lag experiments with young mice, and I'll correlate these results with the stroke infarct volume. I'm also correlating inflammatory status of blood monocytes using Luminex with our metabolomics results. And in my future lab, to fully understand the circadian regulation of immune metabolic response to stroke, I'll use a multi-omics approach to integrate transcriptomics and metabolomics to understand common pathways that are impacted after stroke in both control and jet lag mice. I will also determine if restoring NAD levels, for instance, by nicotinamide riboside, a precursor of NAD, would improve immune response to stroke and ultimately improve outcome. All of my experiments so far has been conducted during the subjective nighttime of the mice, and I'll be repeating these experiments during the subjective daytime of the mice to determine if time of stroke occurrence has an effect on the immune metabolic response to stroke. And with that, I would like to thank my mentor, Kati Andreasen, my collaborators, Melanie McReynolds from Penn State, Ludmila and Alexandrova and Alice Chen here at Stanford, and everyone in our lab, as well as our funding sources. And thank you. I will take any questions.
Thank you, Chinyere. It's fascinating. It's a good lesson that I really should stop staring at my phone at 2 a.m. On a regular basis. So I have, because you have such a powerful phenotyping system, and you use mice as a model organism, I was wondering whether you plan to utilize mouse genetics, for example, some of the clock mutants, and also whether there are ways to get to maybe the cellular specificity of these pretty dramatic circadian effects. That I know all cell types have a clock, but you have been focusing on the monocytes, which seem to have a really strong effect. Can you do something like a adaptive transfer of young monocytes or something like that to kind of parse out the different cell types?
Yes. So right now we're using blood monocytes because they're like the first responders, including neutrophils, that get into the brain after stroke and actually contributes dramatically to the stroke injury. And one of the things that we're going to be doing is repeating these experiments where we have specifically knocked out BMAL, which is the most important core clock protein, where if you knock it out, you lose all rhythmicity. And so we're repeating these experiments in a myeloid-specific BMAL knockout. So you'll have BMAL deleted in neutrophils, monocytes, all myeloid cells, including the microglia and the brain, and seeing how that affects in the immune response to stroke.
Yeah, that perfectly answers my question. That was pretty much what I was getting at. Do you like is a lyse-M Cree, and with the clock mutant, and that is BMAL in your case.
Yes.
Super interesting. And I think I didn't see any questions in the chat yet. We also should move on to our last speaker. Thank you again, Chinyere, for a really interesting talk. And our final and fourth speaker today is Dr. Shira Weingarten-Gabbay. Shira did her PhD at the Weizmann Institute of Science and is currently a joint postdoc in Dr. Charles Rice's lab at Rockefeller and Dr. Pardeep Sabeti's lab at the Broad Institute. Shira is a recipient of the Human Frontiers Science Fellowship. And she is going to tell us about some really novel, interesting non-canonical open reading frames that she discovered in viral genomes. So please take away.
Thank you. Thank you so much for everyone. And thank you for the kind introduction. So my name is Shira and I'm a joint postdoc in the lab of Pardeep Sabeti at the Broad Institute and the lab of Charlie Rice at the Rockefeller University. And in my talk today, I hope to convince you why we need to look more thoroughly on viral genomes if we really want to understand better the interaction between viruses and the immune system. So I will start by describing what I think is one of the most sophisticated and elegant systems in our body that facilitate recognition of infected cells by T cells, probably very familiar to many of the audience. So briefly, when viruses enter the cell, the RNA is translated into proteins. These proteins can be processed into small peptides that can be uploaded on the HLA complex and presented on the cell surface. And while T cells recognize the presentation of a foreign peptide through a specific interaction with the TCR receptor, there can be a cascade of events leading to killing the infected cells, and hopefully by doing that, stopping the spread of new virions into neighboring cells. So this important pathway has been thoroughly investigated for many, many years, but mostly under the assumption that the major source for antigen presentation is coming from canonical open reading frame. These are the annotated proteins in viral genomes that we have some assumptions about how they should look like in terms of what is their size and what is their start codon. But experimental measurements in the past decades expose that there is much more going on. There is more translated regions in viral genomes. We call them non-canonical open reading frame, and they're usually shorter, and they can start with non-ATG start codon, and therefore they won't miss. And they can hide in this, what we used to call in non-coding sequences, such as the 5' UTR, or in overlapping reading frames to annotated coding sequence. And when you take this additional source into account, you might significantly increase what you consider for antigen presentation, and potentially there might be major sources for T cell and T-cell vaccine-based peptides. So with that, two of the key questions of my postdoc is what is translated from viral genomes, information that we do not have for many, many viruses, and second, what is presented on the HLA complex. And while developing experimental system to answer these questions more globally for many viruses that I will mention in the second part of my talk, they became highly relevant for one specific virus of interest, which is SARS-CoV-2 about 2 years ago. And I had the pleasure to join the efforts from the lab of Noam Stengen-Assar and the Weizmann Institute to conduct the first translation measurements across SARS-CoV-2 genome. And together we discovered that in addition to the 12 annotated open reading frame, SARS-CoV-2 also encodes 23 non-canonical open reading frame. And over here, you can see all these open reading frame ranked according to their expression levels. And we were quite surprised to see that some of these non-canonical open reading frame exceed the expression of the canonical proteins. So while we don't know the function, it seems that it is important to take them into account when we think about source for antigen presentation. So we were really interested to take an untargeted approach and to see what is presented on the surface of cells that are infected with SARS-CoV-2 in a way that will allow us also to expose potential peptides from non-canonical open reading frame. So we performed the following experiments. We infected cells with SARS-CoV-2. We did it with two different cell lines. And then we allow enough time for proteins to be processed and presented on the HLA complex. We then purified HLA molecule using a specific antibodies and determined the identity of the presented peptide using mass spectrometry. And the key aspect when searching the data of the mass spectrometry was that in addition to searching peptides coming from the canonical open reading frame, we also search against peptides that can come from the non-canonical open reading frame. So this is a summary from this experiment. On the x-axis, you can see the position along SARS-CoV-2 genome. The first track is the canonical open reading frame, and below you can see the position of the non-canonical open reading frame. And in the 4 tracks below, you can see all the positions in which we found peptides that were presented on the HLA complex. And one of the most striking observations was that not only non-canonical open reading frame can participate in antigen presentation, but in fact, they accounted for 25% of the presented peptide. And more specifically, they originated from 2 overlapping open reading frame in the coding region of the spike and the nuclear protein. And it might be even more impressive when we take into account that the small non-canonical open reading frame account for less than 2% of the entire proteome from SARS-CoV-2, yet they were presented occupying 25% of the HLA complexes. So beside the information of having this non-canonical open reading frame generating peptide that can be presented, the key question, especially in the context of vaccine design, is whether they can elicit a T-cell response. So we first studied these questions with the humanized mouse model. These mice expressed the human HLA allele. We injected them with a pool of peptide that we detected in the mass spectrometry. And then after 10 days, we harvested the spleens and tested T-cell response against individual peptides. So what you can see on the y-axis is the T-cell responses that we quantify in an ELISpot assay. Each column here represents a different peptide. You can see the negative controls and the positive controls. And we managed to identify 4 peptides that trigger a significant T-cell response, 3 of them coming from non-canonical open reading frame. But even more exciting was experiments that we moved to do with humans, with patients that recovered from COVID-19 infection. And then we isolated T-cells from these patients and tested them once again against individual peptides, in this case, using a multiplex tetramer assay. So in this heat map, you can see each line represents a different peptide and each column is a different sample from different human. And we were really surprised to see very strong responses against a peptide coming from non-canonical open reading frame 9b. And in fact, these responses exceeded responses that we observed against what was considered at the time the most immunodominant peptide in SARS-CoV-2. One of them is coming from the spike proteome. So with that information and knowing how significant this non-canonical peptide can be against the T-cell response in COVID-19, yet they were completely overlooked in the first 18 months of the pandemic, and they were not integrated in any of the strategies of vaccine designs and T-cell assays. It's kind of like circling back to these 2 questions I mentioned at the beginning of my talk, that we want to have this information for other viruses. And Christine did an amazing job presenting all the emerging and re-emerging viruses. So we want to be better prepared for additional viruses, hopefully having this critical information in hand already, in the next few years and not waiting for the next pandemic. So in the second part, I will describe some efforts that we've been doing, trying to expose what is translated from viral genomes and trying to expose open reading frame for many viruses. So maybe it's a good time to confess that my background is not in virology, and I started work on viruses when I joined my postdoc lab about 5 years ago. But before that, I did a PhD in systems biology. And during my PhD, I studied gene expression regulation, and I developed 2 high-throughput systems that allow quantitative measurements of tens of thousands of synthetic sequences at once, either for transcription regulation or translation regulation. And starting my postdoc, I thought maybe we can use a similar synthetic approach in order to discover open reading frame, not in one or few viruses, but in hundreds of different viruses in one single experiment. And this is the general idea, is to design a synthetic library composed of tens of thousands of sequences representing hundreds of different viruses that are pathogenic to humans. Then to express all of them together in cells and to isolate regions that are directly bound and protected by ribosomes. Then you can use deep sequencing approach to infer for each one of the design sequences, whether it's contained an open reading frame that were translated by ribosomes, and if it does, what is the exact location of this open reading frame within the design sequence. So using this approach, we did a pan-viral library, and we screened almost 700 different viruses, representing 32 different families with more than 4,000 genes. And a key question is whether you can use this synthetic approach in order to detect open reading frame while they're outside the context of native viral infection. So to answer this question, we first look at a set in which we have annotated coding sequences. So this is just one example, one gene from the human CMV virus. And on the x-axis, you can see the position along the design oligo. On the y-axis, you can see the number of ribosome footprints that we detected. And as expected, we can see ribosomes recognize the start codon and accumulation of ribosome in the coding sequence. But we have this information for other genes as well. So we can now aggregate all of them into one meta-gene analysis. And in this case, looking at more than 1,000 genes from the herpes viridae family, we can see that using our assay, we can accurately detect annotated coding sequences. And not only that we can see the accumulation of ribosomes, they're also accumulated in the right reading frame, which is information that we can infer by using the deep sequencing of ribosome-protected fragments. So this is true not only for herpes viruses, we now look at other families that we have in the screen. So these are just examples of 6 additional families. And it's quite remarkable that although these viruses have different life cycle, they have different nucleic acids, in the synthetic systems, we can successfully capture the annotated coding sequences in each one of these families. So we're really encouraged by these results, and now we want to ask the interesting questions, right? What are the novel or non-canonical open reading frame in these genomes? So we took all the data that we generated from this pan-viral library, and then we adapted a protocol in order to infer open reading frame using the ribosome footprint. So very shortly, it takes information about the ribosome footprints from the experiments and how the sequencing reads can align back to codons in the genome. And using this approach, we uncover 7,000 viral open reading frame in more than 3,000 novel non-canonical open reading frame. And it's very exciting time for us. And what I hope to do in the next few years of my independent lab is to use this vast resource of non-canonical open reading frame that we can now have for so many different viruses to understand better how they can play into the interaction between viruses and the immune system. And I will just mention that one of the reasons I think this is an exciting source to look at is that it represents a different playground than the other source for T-cell epitopes, right? Non-canonical open reading frame can be potentially mutated more easily, meaning that maybe viruses can use them as a mechanism to evade the immune response. But even beyond looking at the interaction and how this open reading frame play a role in the antigen presentation pathway, non-canonical open reading frame were also shown to play a role in viral gene expression regulation. So specifically, upstream open reading frame can occupy the ribosome in a way that they will interfere with translation of the main coding sequence. And in our screen, we expose hundreds of new upstream open reading frame. And I'm very excited about looking what the role in regulating gene expression in viruses. And the third thing is that we have additional source for polypeptide that are translated in infected cells, and some of them may be functional and play a role in the viral life cycle. But beyond this plan for the first few years, I want to show you maybe a more general vision, and what I hope to do down the road for the long term is that I will be passionate about combining these two worlds together, my experience and training from my PhD in systems biology and my training in virology, in order to design new methods to ask more systematically about different aspects of the viral life cycle. So the idea is to maybe if we can transition from looking at one gene and one virus at a time into many genes in many viruses will help us to expose some mutual principles in viruses that we cannot see whenever we just look at one virus. So developing systematic approaches to tackle different aspects of the viral life cycle, maybe we can cover new insights into the complex biology of viruses. So with that, I'll end. I want to thank all of you. I want to really thank my two amazing mentors, Pardisa Betty and Charlie Wise, that provide so different perspectives on this field of virology, from viral genomics from one hand and fundamental virology from the other. From the Sabeti lab, Matt Bauer is a talented grad student that worked with me on the development of the massively parallel ribosome profiling to discover open reading frame. And Leah Perlman is a technician that now pursuing master in public health that worked with me on the HLA purification from cells that are infected with high contaminant viruses. Muhsin Saeed did the SARS-CoV-2 work in his lab in Boston University. Susan and Jen from the proteomic platform did the mass spectrometry analysis, and Sissy from New York Ryan's lab did HLA predictions, and many other groups help us with the TS and T-cell assays and other aspects of this work. Huge thanks to the Leading Edge. I'm really honored to be part of this supportive environment, and these are my postdoc fellowship, and thank you so much for listening.
Thank you, Shira. That's super intriguing. We have a question in the chat. Jeanette asks, are the non-canonical ORFs less stable or more prone to proteasomal degradation? And might that account for their preferential presentation?
Yeah, so that's really, it's a great point. We haven't tested their stability. In general, they are shorter. And if they're not folded into a more complex protein structures, we think that they might be degraded more quickly. And they're also generated, some of them at least generated very, very quickly. We found also in the study of the SARS-CoV-2 that there is some connections. Viral proteins that are translated earlier in infection also tend to be presented on the HLA complex more frequently. And we found, for example, that open reading from 9b is translated as early as 3 hours post-infection. So we think all of these parameters will probably help them end up maybe even more preferentially on the HLA complex. And in general, there were many studies that showed that there is some linkage between translation and presentation. When people inhibited translation, they stopped the process in which peptides are loaded in the endoplasmic reticulum on the HLA molecule. And it kind of like suggests that if you have all these resources quickly translated, they don't necessarily have a function, they don't necessarily fold into a protein with high half-life, with very long half-life. Maybe they can end up more quickly on the cell surface. And it's also alluding to a theory, although by a different mechanism, that was suggested by Jonathan Yaldell and other colleagues, that called the DRIP theory, the defective ribosomal product, suggesting that if you really want to identify infected cells, you want to do it early in infection. Right? It doesn't help you if you now see cells that are infected with influenza, let's say 24 hours post-infection or 12 hours post-infection. You really want to identify, to have these viral peptides presented, let's say an hour, 2 hours post-infection. So the DRIPS theory suggests that there is a reduction in translation due to interference, and then these immature polypeptides will end up degrading more quickly and presented. So we think that non-canonical open reading frame can act in a similar mechanism, although I think we just need to know more about it. And maybe the first step is exposing these non-canonical open reading frame.
Jessica.
That was fascinating, really fabulous talk and such a cool ribosome profiling technology. So I'm by no means an expert on viruses, but what I do know is that with such a small genome, it seems like even these non-canonical protein products must have some sort of function. Do you have any hints as to whether those these peptides that you're seeing might play functional roles, or do you think it's maybe just a strategy for rapid evolution? Like they have these products that could perhaps evolve more rapidly than the ORFs that play roles in replication. Anyway.
Yeah, no, I think these are all amazing questions. And I think kind of like, I don't know the answer to many of them. You can ask me what will be the dream experiment for me or the dream information if for each one of these non-canonical or for each one of these peptides coming from non-canonical, we can know both what is the potential in antigen presentation, then how likely it will be recognized by T cells, and then what is the effect of mutation in this region on fitness. And if we'll have all this information together, I think that we can start to understand their function. Some of these non-canonical open reading frame also with SARS-CoV-2 are conserved. Some of them have some annotated domain, and we actually think they have function. Others do not. In general, they have more mutation than canonical one. And it is kind of like, I think that maybe even one of their function can be potentially, if they are participating in viral immune evasion, then their function will be to serve as a decoy peptide that can acquire a lot of mutation, keep evading recognition by T cells. And this might be the function by itself, right? But maybe there are other functions as well that I think we can just start to understand now that we expose more of this non-canonical.
Thank you. I'll ask one last question before we end the session. Some of the human genomes contain some viral genomes from past ancient infections too, right? Like, yeah. Do you have any information on like what proportion of that has translated non-canonical ORFs, and whether that has anything to do with, say, like autoimmune diseases?
Yeah, so it wasn't, I've never looked at it more systematically, but there are amazing works of people combining ribosome profilings and doing it on various cell lines, including cancer, and trying to understand better whether viral sequences can participate in antigen, endogenous viral sequences can participate in antigen presentation. And the answer is yes, they can generate proteins that can be presented. But if it's constantly presented or expressed in the cells, this is something that you also expect that will be expressed in the thymus as well. And maybe there is already selection against them. So you don't expect them necessarily to be a good target for T cells. What is interesting in what other people are doing in their space is trying to see if these viral sequences can have differential expression in cancer cells, for example. And in this case, they can serve as good neoantigen targets because they're not expressed in other cells in our body. There wasn't selection against them in the thymus. So I know a few works of people trying to look in these endogenous viral elements as potential targets for cancer therapy, which is very exciting.
Yeah, it's super interesting, and as you said, largely overlooked. With that, let's end this really exciting session and thank all of our speakers for brilliant talks. And before we end, as a reminder, please tune back in on Friday at the same time, noon Eastern time, for our third neuroscience session. And goodbye, everyone.
Thank you.